Hey everybody, this is Crystal Ivy, your instructor, and I'm going to go over chapter 4 with y'all today. So chapter 4 covers cells. I want to go ahead and forewarn you, I have a 2.5-year-old who is not in bed yet. So hopefully we won't have any distractions. He's in there watching The Land Before Time. Littlefoot, I don't know if y'all remember that. That's something I used to watch when I was a kid, and now he's like in love with it. But hopefully we'll be able to get through this while he's watching that movie. So chapter 4 again covers cells. And so 4.1 is cells under the microscope. So cells are studied using microscopes because they're really small. Cells are not able to be seen with the naked eye. They're very diverse. They look very different from cell type to cell type, but they're all very small. Each type of cell in our body is specialized for a particular function, which shouldn't surprise you because if you've been watching these narrations and reading in your text, you've noticed I harp on this all the time about how structure and function go hand in hand. The structure of a cell is structured in such a way that allows it to do its function. No different than an entire organism. A frog has really long, powerful legs that allows it to leap really high. They're structured in that way because they serve a function for that frog, just like our cells serve a function for our body. One of the most common methods of visualizing cells under a microscope is with what we call a light microscope. And when you do lab, you will be using a light microscope, a compound light microscope in lab. This type of microscope was first invented around the 17th century, which is a long time ago, it seems. And that first microscope was very primitive, but we have built on that to make them bigger and better than they were back in the 17th century. But they use the properties of light to enable us to visualize organisms or cells at a much higher magnification than with just the naked eye. However, we are limited by the properties of light, and so to evade or circumvent the issue of the properties of light, the electron microscope was invented in the 30s. And so since it doesn't use the properties of light, it doesn't use light, it uses electrons, we don't have those same limitations. These electron microscopes are extremely powerful, way more powerful than our light microscopes. Light microscopes, they go up to, maybe 1,000x is pretty standard. Some maybe 2,000x, but 1,000x is really standard. And it's actually what we have in our lab on campus. Electron microscopes, I believe it's some of them I've seen are 100,000x or more. All right, so they're very powerful, very, very expensive as well. Think these electron microscopes are like $500,000, probably sometimes more than that. Very expensive, but very powerful. So here's some pictures showing you some demonstrations of cells being visualized using microscopy. So this guy here, this is a cell showing lots of different organelles within it. You can see the nucleus is here. We'll talk about what that is in a little bit. It's a very prominent organelle, and lots of other ones as well. You see those in green. And the type of the type of microscope that took this picture is called a TEM. That's a transmission electron microscope. And they call it a transmission electron microscope because it literally transmitted. It went through the cell to take a picture of the inside of that cell. Here's a picture of a scientist using that said electron microscope. Over here, we have an SEM. That's a scanning. I'm just going to use my chemistry symbol for electron. Scanning electron microscope. And so scanning electron microscopes still use electrons, but they scan the outside of the organism. So that's why this guy here looks three-dimensional, because those electrons scanned the outside of it and took a picture of that. Whereas again, transmission transmitted to the inside of the cell and took a picture of a plane within it. So that's why it looks two-dimensional instead. Over here, we have a light microscopy image of a cell that we call a euglena. A euglena is a protist. It's a single cellular organism that's in the kingdom Protista. It's a protist. Remember I said from chapter 1, that's kind of like the junk drawer of biology because it's not a plant, it's not an animal, it's not a fungus, so it's a protist. But you can see it's a single-celled organism, and it is under a light microscope. Light microscopes, again, use light. This is at 470x, so pretty powerful microscope, but not nearly as powerful as these other two. And here he comes. Shh. Mommy working. Okay, so next we would need to talk about the size of cells. So we already said they're really small, and they are, but why? And how small are the next questions we're going to answer. So if you look at this graphic here, it shows you different various objects in order of size, starting over here at the left, which are really small things, and over here at the right, which are very, very large things. And it also shows you how we can visualize them. All right, so atoms are pretty much right on the edge of being able to be seen with electron microscope. We do have some more powerful microscopes that the textbook doesn't mention than the electron microscope that does enable us to see atoms. But the electron microscope can definitely see things like amino acids. And if you'll recall, amino acids from the last chapter are the building blocks of proteins, and they're really, really tiny. So these amino acids are only 1 nanometer, really small. I put into one of the folders in the lessons tab on week, I believe it was either 1 or 2, but some optional items in lab where you could work on your metric units like meters and liters and centimeters, millimeters, grams, temperature as well, which is Celsius. Some of y'all may be pretty familiar with our metric system of units because you might have learned about it in previous classes or in high school or even middle school. I know they start teaching it sometimes even in elementary school at this point. So I didn't make it required, but it is there for you. It is optional. If you need to familiarize yourself with this content, then please go do so. Please go do so if you feel that it will help you. But 1 nanometer is very, 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 very small. Proteins, you're looking at around 5 to 10 nanometers. Viruses, lovely things that can make us sick sometimes. They're going to range between typically 10 and 100 nanometers. All right, next we're getting into organelles. We'll talk about what these are, what organelles are today in this lecture, but they're about 1 micrometer. Notice we're in a different unit now. Micro, versus nano. All right, so chloroplasts, they're an organelle, they're about a nanometer. Most bacteria fall here within I said nano, I meant to say micro, between 1 micrometer and 10 micrometers. And cells themselves, plant and animal cells, fall typically within 10 and 100 micrometers. So going a little bit larger, we see the human egg. So believe it or not, that's just on the cups of where we will see things with a light microscope. The electron microscope. We're going to need definitely a light microscope to view anything from here, the frog egg and the human egg, all the way to the chloroplast, to organelles in general. Once we get smaller than that, we're going to need an electron microscope. Now, we could use an electron microscope to view these things here. It just wouldn't be very practical. Why buy something so expensive whenever we can use a light microscope? Moving forward, the frog egg, now we have an ant. So ants are definitely able to be seen with the human unaided eye. All right, we can actually see a frog egg too, and kind of a human egg. I mean, barely. They're very, very small. But again, this shows you a few more items: a mouse, a human, and a whale. Obviously, those items you wouldn't need a microscope, but this shows you a breakdown of the various items that you're familiar with and where they fall on this scale of size and what type of microscope will be beneficial to see those things. Okay, So now that you know about what size cells are in general, why are they so small? Why? So cells are so small because one thing that's critical for them to be alive is that they are capable of transporting materials. Okay, and if a cell is really, really large, then that transport is going to take a very long time. Same goes with exchange. Our cells are able to take stuff in from the environment, and they also have to get rid of stuff like waste. For example, carbon dioxide is a waste product in cells, and then we breathe it out. If that took a really long time, the toxicity of it would be overwhelming for the cells, and it would cause cell death. Okay, so I just want to draw a little picture up here. So here's cell number 1, and here's cell number 2. All right, so if we were to, and of course this is hypothetical, if we were to measure the distance between, across cell 1, measure the diameter there, and compare it to cell 2, this cell here is obviously very big compared to this one. So if you can imagine a substance trying to travel across this cell from one point, we'll call this point A, all the way to over here to point B, it's not going to take a long time for it to get from point A to point B compared to how long it would take to get to point B from point A in cell number 2. So that's why cells are so small. They need to have exchange and transport of materials into, out of, and even within the cell itself. And if they are very big, then that's just going to take too long to be compatible with life. We call this concept cells needing a very high surface area to volume ratio. Now let's think about what that means. Because basically what it's saying in fancy terms is that small cells are better at exchanging and transporting material. But the fancy way of saying it is this cell here has a higher surface area to volume ratio compared to that one. So what does that mean? Area, if you were just look at the definition, take yourself back to math from grade school. Area is a measure of how much space there is on a flat surface. And of course, this is a circle, so you need to imagine this is three-dimensional. So it would be a sphere. So it's not technically flat, but you can still find the area of that circle. I actually just went over this with one of my stepchildren. I had to teach him how to find areas of circles and spheres because they're of course out of school due to COVID-19, so that was fun, me teaching math. But anyway, area is the measure of how much space there is on a surface. It doesn't necessarily need to be flat. So, and I'm going to try to draw this the best I can, so bear with me. I'm trying to draw a rectangular prism. I don't think I'm doing too terrible. So if we were to take the area of this space here in the front, the one in the back, and of all 4 sides and added it together, that would be the surface area. It's the area of every single surface within that shape. Now volume is the amount of space something takes up. So if I were to fill this cube or prism, whatever it is, with water, then we'd be trying to figure out how much volume is in there. All right, so you can kind of imagine it as like an aquarium. We could take the surface area by calculating the area of each space, and then if we were to be measuring the amount of liquid that it holds, or that would be the amount, the volume that that object occupies. All right, so this picture kind of elaborates on cell size and surface area to volume ratios. So what it's demonstrating is 3 different possibilities. The first one here is representing a single cell, which is really small. This one is representing a single cell that's really big. And this column here is representing lots of little squares or cells that are the same size as this one, but fill up the same volume as the second one here. So let's calculate the total surface area, volume, and ratio for each one. So this one, the total surface area for this guy is going to be calculated by height times width times the number of sides times the number of boxes. So to get that, we have 1 times 1 times 1. Oops. Nope, 6. 1 times 1 times 6, because that's the number of sides, times 1. All right. The height, it says it's 1. The width, since it's a square, it's the same. The number of sides, if you count them up, you only see 3, but there's 1 underneath, 1 on the back, and 1 over here. That gives you a total of 6, and there's only 1 box. So 1 times 1 times 6 times 1. That gives you 6. If we calculate the volume, we have the height, which is 1, times the width, which is also 1, times the length, which is also 1, and times the number of boxes, which is also 1, which gives us 1. 1 times 1 times 1 times 1 is 1. Okay, so now the surface area to volume ratio is simply just taking the surface area and setting it in a ratio format to the volume, which would be 6:1. Now that is the same thing as saying 6/1, which gives you 6. So our surface area to volume ratio for column 1 for this one is 6. Let's move to the next one. So here we have a giant cell. The height is 5. The width is 5. The number of sides is 6. And the number of boxes is 1. So if we calculate that, 5 times 5 times 6 times 1, we get 150. Now for the volume. The height is 5 times the width, which is the next thing, which is 5 times the length. Which is 5. And now by the number of boxes, which is 1. So if you calculate 5 times 5 times 5 times 1, you'll get 125. So if we were to set that up to as a ratio, the surface area, 150, set it to 125, it's the same thing as saying 150 divided by 125. If you calculate that, you'll get 1.2. All right, so now moving here to the last one. The total surface area for all of these collectively is basically the same thing as taking the surface area of each one of those, which we already did, and then multiplying it by the number of boxes that we have. So again, the height times the width times the sides. We know the height is 1. We know the width is 1. We know the number of sides is 6. Now we need the number of boxes. And so there is 1, 2, 3, 4, 5 boxes. And then 1, 2, 3, 4, 5 boxes going down. And 1, 2, 3, 4, 5 boxes going back. So that means that we have a total then of 5 times 5 times 5 boxes, I believe. Let me check my math here. Let's see, that gives me 125 boxes. So where's my pen? Let me write that down. Okay, so let's do the math here. 1 times 1, of course, is 1, times 6, times 125. That gives me 750. Volume, the height, the width, and the length all multiplied together times the number of boxes. So the height is 1 times the width is 1 times the length is 1 times 125. That gives us 125 for our volume. So for the surface area then, we would set 750 colon 125. And again, that's the same thing as saying 750 divided by 125, which gives us 6. All right, so I hope that will help give you all a better understanding of surface area to volume ratios. So now the last thing to kind of piece the puzzle together is that as surface area to volume, or as your surface area to volume ratio increases, then that means it's going to be better for the cells. Right, so as surface area increases, in these three examples, the volume, or these two examples, the volume stayed constant. The volume within all of these little tiny squares collectively is the same as this, but the surface area is greater in the last column, which means that this option between A, B, and C is providing the highest surface area per volume. So the highest surface area per volume ratio yields smaller cells that have lots of volume that has smaller cells with lots of surface area compared to this one, which means because they are so small, they are able to transport materials in and out and even within them much quicker than they would if it was in this large cell. It's kind of like imagining, if you're, driving a car, if you're a really, really tiny little micro vehicle and you're driving within this little cell, it's going to take you very little time to cross this cell, this one tiny one, versus how long it would take you to cross that one. Okay, so how is this relevant? So this is relevant in several areas of the human body, but I'm just going to really mention maybe one or two. The one that's in the PowerPoint is the microvilli and the villi within the intestinal tract. So your intestines, or your GI tract, your gastrointestinal tract, is basically everything from your mouth all the way down to the anus. So it starts with your mouth, it goes down to the pharynx, and then the esophagus, and then to the stomach. The stomach is pictured here. So this is your esophagus. Here we have the stomach, and then this is the beginning of the small intestines. So all the stuff here you're seeing is intestines. Here's the large intestine that surrounds it, the colon, and finally the anus down here. So it's basically, really one large tube. It has a large expanded area here, which is the stomach, but really it's just a big long tube that runs from your mouth to your anus. Now, if you were to take a cross section of your intestines, which means if you were to cut it and look down it just like this, like you're looking down a water hose, this hole here is where food passes through. And if you notice, on the edges, it's not smooth. And that's because if you were to zoom in, what you're actually looking at are foldings within the intestinal lining. Those foldings are called villi, all right, and it's shown even bigger here. So this little small section, they blew up really large. It looks like little fingers. So it's not smooth. It has all these little finger-like projections. Now, why might that be? Why might it be that these intestines, which purpose, by the way, is to absorb the food that you eat for nutrients for your body, why might they have all these finger-like projections? And the answer is that it increases the surface area to volume ratio. Okay, so again, imagine if your intestines were just a big long tube that didn't have all these foldings. I'm just going to draw it like this, like a straw, basically. Think about it. If you're driving a car, if you're going to drive from point A to point B, then that distance is, let's say hypothetically, 10 feet. Now let's say, in our second example, that this wasn't a straight road. It had lots of hills and valleys. Well, now you're not just driving 10 feet. you're driving the distance up the mountain, down the mountain, up the mountain, down the mountain, up the mountain, down the mountain, and so on and so forth. So it's actually much longer than 10 feet whenever you take all that into consideration. But if you're on the ground without the mountains and everything, it still just seems like 10 feet. Okay, so we're increasing the surface area. So we'd effectively be increasing, with my example of the car, we would be increasing the amount of tract we would cover, the amount of road we would cover, even though we still only traveled in a two-dimensional mindset 10 feet. Now, more so, these cells that you see here, these little tiny boxes all over these villi, these cells have microvilli at the top. So you see these little hairs, they're even more extensions. So that increases the surface area to volume ratio even more. And without this adaptation, your intestines would have to be hundreds of meters long in order for it to serve its purpose in your body. But because of all of these adaptations, these villi and these microvilli on the cells, they don't have to be hundreds of meters long and are capable of fitting within our stomachs. All right, so 4.2 is the plasma membrane. The plasma membrane is common to all cells, which means that all cells have a plasma membrane. The plasma membrane is what separates the inside of the cell from the outside of the cell. So you can kind of think of it like a water balloon. If you were to fill up a water balloon, that rubber or latex that makes up the balloon itself would be equivalent to the plasma membrane. It's what holds the contents of that water balloon within the water balloon. Same thing with the plasma membrane. It holds everything inside. It's made up of a phospholipid bilayer, which we discussed in a previous chapter. So if you'll recall, our phospholipids are an example of a lipid. So they have a structure that looks just like this. They have a polar head, which you'll recall is what? Hydrophilic or hydrophobic? Polar head. It is hydrophilic, which means it likes water. And then it has a nonpolar tail, which is made up of two fatty acids. You can see one here and the second one there. The nonpolar tail is hydrophobic, which means it does not like water. Remember, polar things like other polar things. Polar things don't mix with nonpolar things and vice versa. So nonpolar things like nonpolar things and they don't like polar things. That's why if you were to take a glass of water and put oil in it and shake it up, it's never going to really mix. If you leave it on the counter for a little while, you'll begin to see the oil separate from the water. You can do that little experiment if you'd like to. If you have any baby oil or something laying around, mix it with some water and see what happens. Okay, so that phospholipid bilayer is demonstrated down here. So if we break this down, we said it's made up of phospholipids and it is a bilayer. That means it has two layers of phospholipids, and it does. So you can see the first layer is on top. You can see all of the little red balls are representing the polar heads of all of the phospholipids. And then the fatty acid tails are here. All right, and then you see our second layer of phospholipids on the bottom. Again, polar heads are the little red balls, and then the fatty acid tails are in yellow. Now, why is it orienting itself this way, where the polar heads are on the top and the bottom, and then all the fatty acids are sandwiched in between? Why? The reason is because the outside of the cell is mostly water. The inside of the cell, which is over here on this side of the plasma membrane, is mostly, again, water. So again, these phospholipid heads, the polar heads, like water. So they're facing water, right? These heads over here are also facing water. the tails are turned in towards each other. They're not going to face the water because they're hydrophobic. They don't like it. So they do something different. They face one another because these non-polar hydrophobic tails are attracted to these non-polar hydrophobic tails. All right. Now, something else you should know about the plasma membrane is it is selectively permeable. What does that mean? Selectively means that it's picky, and permeable means that something can be moved in or out of. So our plasma membrane is picky about what it allows to pass through the plasma membrane. All right, and so we don't want just anything and everything to get in our cells. So for example, we don't want excessive amounts of sodium to get inside of our cells because that would throw off the balance. It needs to have a normal reference range in order to be compatible with life. We can't allow too much hydrogen ions to get into the cell because that would turn the cell's insides acidic, which may not be compatible with life. We don't want viruses and other pathogens, bacteria, etc., to get into our cells because it can make us sick. So our cells are considered selectively permeable. It picks and chooses what it will allow in and out and what it won't. Now, that's not to say that sometimes our cells don't let in or out things that it shouldn't. So the example I gave you with the immune system cells, allowing in bacteria or viruses, sometimes those things do trick our cells into letting them in. But typically, the point is it's selectively permeable. It doesn't just let anything and everything in and out. It's not a free-for-all in there. Another term you may hear in our textbook is the fluid mosaic model. And so we call the plasma membrane an example of a fluid mosaic model because, for one, it's fluid. And when I say fluid, I don't mean a liquid. I mean it's flexible. It has a lot of fluidity to it. The reasons why it is very fluid like that is because it contains lipids called cholesterol. So if you recall from last chapter, cholesterols are examples of lipids. So those are seen in this picture here, 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 and there. Those are cholesterol molecules, and it helps give the cell flexibility. Now, it's called a mosaic because there's a pattern to the proteins that are embedded into it. So if you'll see this bluey purple thing here that's embedded into the plasma membrane, and this one here, and that one, and that, those are proteins. And there is a pattern to what kind of proteins are on a cell based on its cell type. So, for example, this we'll say is a T helper cell. It's a cell that is of the immune system. If you've ever taken AMP, you probably know a lot about those. We won't go into too much detail here, but for example, these T helper cells have little protein receptors on them we call them CD4 receptors. And so that's specific to this cell type. Other types of cells, like a skin cell, or even other cells of the immune system, like B cells, they don't have, I put this is my symbol for no. They don't have CD4 receptors. They have their own specific set of proteins embedded into their membrane. Okay, so again, it's called a fluid mosaic model because it's flexible due to the cholesterols in it, and it's called a mosaic because it has a pattern of various types of proteins that are embedded into the plasma membrane that is based on cell type. Okay, so those proteins that are in the plasma membrane can come of different varieties. There's several of them. We'll discuss quite a few of them. The first one are channel proteins. And channel proteins is just like it sounds. It serves as a channel for stuff to move. It forms a tunnel for very specific molecules. So, for example, aquaporins. And if you break that down, Probably can guess what it means, right? Aqua, pour in. Aqua meaning water, and pour meaning it has a pore, a hole. So an aquaporin is a channel, a hole with a tunnel that allows for water to pass through. And so aquaporins are proteins. Aquaporins are channel proteins that are specific for water. Transport proteins are the second type. They are involved in passage of molecules or ions through the membrane, and sometimes it requires energy. So one example would be the sodium-potassium pump. This particular image is not of the sodium-potassium pump specifically, because what the sodium-potassium pump does is it pumps sodium and potassium in and out of the cell. It'll pump sodium in and potassium out against their concentration gradients. We'll talk about what that means later. But in general, that example does require energy. And in the image here, all it is showing you is just an example of where things can be loaded onto a protein. These little green balls are representing a molecule or an ion that can be loaded onto a protein and then moved to the other side of the cell. It's transporting it from one point to the next. Note that it's different even than the previous example we gave. They serve similar functions, but they don't do it the same way. Here, this is the one before with a channel protein. It literally is just a channel. It is a tunnel where things that are able to pass through specifically. Aquaporins was one example for water. Here, we do not see a tunnel. It is going to allow the passage of molecules or ions, but it does not serve as just as a tunnel. It has to be taken by the protein and basically spit out on the other side. Next, cell recognition proteins. So if you look back, I'm going to jump back a few slides. You see this guy back here? I just called him a protein before, but now I'm going to elaborate because he's not really just a protein. He's a glycoprotein. That's special. He's a glycoprotein because he's a protein that has a carbohydrate chain attached. This green here that has several green little hexagon shapes, that's a carbohydrate hanging off of it. So that's why we call it a glycoprotein. Glyco, think, glycogen, glucose has something to do with sugars. Simple sugar chains are carbohydrates. So it's a carbohydrate chain attached to a protein, hence the name glycoprotein. Okay, so here's the picture of it. You can see your protein and the carbohydrate chain here, hence the term glycoproteins. They're sugars bound to proteins. But they enable our body to distinguish between our own cells and cells of other organisms. All right, cell recognition proteins. What does this mean? Recognition. When you have the term recognition, what do you think? Recognize. When you recognize someone, that means, oh, hey, you know them. They're familiar to you. So these particular types of proteins, cell recognition proteins, distinguish between our own cells and cells that aren't our own cells. So a great example would be the immune system. So our white blood cells, which are part of the immune system, recognize glycoproteins on other cells that are either ours and are good, or they'll recognize something that's foreign to them. Like, ooh, I don't know what this is. We need to destroy it. So that's cell recognition. Next are receptor proteins. So receptor proteins involve signaling molecules. So in this case, this would be the signaling molecule. This would be the protein. And the signaling molecule will bind to the protein. You can see that it has a particular shape that fits the protein receptor site. Receptor, it's receiving it, it's receptive. And so again, once the signaling molecule is bound to the protein, it will cause a cellular response. Typically, that response is a very long cascade of events. So we won't go into any one particular one in a lot of detail because there's just so much, a lot of different chemical reactions that occur back to back to back that lead to some kind of result. So again, receptor proteins lead to a cellular response. That's what you need to know. A great example is insulin. So insulin is an example of a signaling molecule. And that signaling molecule will bind to a receptor site for insulin on the liver, in liver cells. And the response here would be a cascade of events that causes glucose to be stored in the liver as glycogen. So if you recall back to our chapters on chemistry, we discussed how various different carbohydrates. So we talked about glycogen and how that is how glucose is stored in the liver. Glucose is strung together. Lots of glucose molecules are strung together, forming covalent bonds to make a really long chain of them, and that is glycogen. It's stored in the liver. In plants, it's the same thing, but it's a little bit different structure, and it's called starch, and it's stored in plants. So here, in this case, the whole reason that happens is in response to insulin binding to a receptor protein that then tells the liver to store glucose in the form of glycogen. So the key here, receptor proteins, you get a cellular response. Something happens because of that binding. Next, enzymatic proteins. So they participate in metabolic reactions. And we'll talk about metabolic reactions at another time. But essentially, this shows you the protein. And it has an active site here where these potential signaling molecules or substrates is what we'll actually call them later when it comes to enzymatic reactions. But they can bind, and then it can alter that that substrate to make it something different. And again, we'll get to that later. But do know that enzymatic proteins participate in metabolic reactions. Think metabolism. All right, next, junction proteins. Junction proteins form junctions between cells. Who would have thunk that? So what is a junction? It's just a place where something meets, right? So here would be, this has a small excerpt showing plasma membrane from cell 1. And then we have plasma membrane at the bottom for cell 2. And I'm just going to write 2 for simplicity. But that's showing you 2 cells. It's small excerpts, but if you can imagine it, here would be, if I drew the whole thing, cell 1. And this would represent all the plasma membrane, or all of the phospholipids all the way around it. And same thing here. But they're just showing you small bits for simplicity. Now we see protein here and a protein there, and they're touching. They're keeping these 2 cells together. So a great example is there's lots of junction proteins between the cells that make up your bladder. Because what happens whenever you go a long period of time without using the bathroom? Your bladder is filling up with urine. It's expanding that organ. But the cells don't ever just, explode, or, they don't ever just pop. The bladder doesn't just pop because you're full of urine. Because these junction proteins keep the cells together. So even as it fills, it's still going to remain intact. It will expand and get bigger, but it's never just going to pop. All right, so that's because of these junction proteins. Okay, 4.3 discusses cell theory and the 2 types of cells. So cell theory, which as we know now from chapter 1, theories are really important. There's not really a whole lot of questioning theories, even though we might talk about theories in layman's terms as not being a big deal in science. Scientific theories are majorly a big deal. So the 2 statements that the cell theory states is that all organisms are composed of cells. Remember, cells are the basic unit of living things. And all cells come only from pre-existing cells. So basically, you know, their cells are not just going to poof, pop up out of nowhere. They had to come from a pre-existing cell. That might make you think, well, where did the first cell come from? It's a good question. Look it up. All right, so all cells also have these three things. One, a plasma membrane. All cells have that. It doesn't matter if it's a plant cell, an animal cell, a protist cell, or a bacterial cell. All of them have a plasma membrane made up of phospholipids. Number two, they all have cytoplasm. This is a new term. Cytoplasm is the liquid within the cell. So if you were to imagine our water balloon filling up, and we said that that latex or rubber that makes up a water balloon, we said that's the plasma membrane, then the water would be the cytoplasm. That's where all the chemical reactions occur in cells. And then number three, they all have genetic material for growth and reproduction. DNA, typically. All right, so two main types of cells. There's two main types of cells, and they're based on the organization of genetic material, of DNA. Prokaryotic cells, they lack a membrane-bound nucleus where the DNA is stored within eukaryotic cells. All right, so prokaryotic cells, if you were to break this down, pro means before, karyon means nucleus. So these are cells, actually, they're very primitive, that came before eukaryotic cells, and they don't have a nucleus. That's what you need to know. Prokaryotic cells lack a membrane-bound nucleus. Eukaryotic cells, on the other hand, have a nucleus that houses the DNA. So let's go into a little bit more detail on the structures of that are found within prokaryotic cells. So prokaryotic cells, we already said, they do not have a nucleus. These cell types are found in two of the three domains that we discussed in chapter 1. Prokaryotic cells are bacterial cells and cells that are in domain archaea, which is very similar to bacteria. Generally, prokaryotic cells are going to be a lot smaller and simpler than eukaryotic cells, and that allows them to be able to reproduce very quickly and effectively, and they're actually a very extremely successful group of organisms. There are so many more different bacteria and so many bacteria just in general than there are individuals of other species like animals on the planet, even within you. So there are more bacteria cells in your body than there are human cells, you cells. There are more bacteria in your body than there are cells that are specifically yours. It's kind of crazy. Now, some bacteria are well known because they can cause disease. However, only 1% of bacteria cause disease. The rest of them don't cause any problems or can actually be very beneficial. They may have beneficial roles in our bodies, for example. They may have beneficial roles in the environment, breaking down stuff to help recycle nutrients in the soil, for example. Some are even used to manufacture chemicals. For example, insulin. If you had to have a synthetic form of insulin or a non-human form, oftentimes bacteria are used to produce human insulin in a laboratory setting. Several years back, they used to use, I believe, pig insulin for patients who had diabetes, but now we can harvest human insulin using genetic engineering using bacterial cells. Some can be even used to make food, drugs, etc. Okay, so let's talk about some structures that make up bacterial cells. Of course, the plasma membrane. We already discussed that. The cell wall. So some eukaryotic cells have a cell wall as well, but not all of them. But bacterial cells, typically all of them have a cell wall. Not all of them, but for the vast majority, they do. But the cell wall helps maintain the structure. It lies right underneath the plasma membrane. Just think of it like an extra layer of like protection and to help the infrastructure of the cell. The cytoplasm is the fluid. I meant to put within the cell. I've made a typo here. Within the cell. Sometimes bacteria have a capsule. A capsule is a little gooey protective layer that helps prevent the bacteria from getting attacked by other bacteria or chemicals, or be protected from sunlight or even the human body and our immune system. DNA. So DNA is housed inside of our bacterial cell in what we call the nucleoid. Notice that this is not the nucleus. Remember, that's my signal for not. It does not have a nucleus. It has a nucleoid. And the nucleoid is just a region. It's just an area where the DNA kind of free flows around. But it is not encapsulated by a membrane. When I say membrane, I mean like a sac. It's not encapsulated in a sac like it is in eukaryotic cells. Next, ribosomes. These are the site of protein synthesis. Super duper important. They are critical to life. Next, there's a couple of appendages that bacterial cells can have. Not all cells are going to have these appendages. Some will and some won't. Flagella. One of my favorite words. Flagella. They are basically a long whip-like tail. So if here's my bacterial cell, they'll have that long whip-like tail, and that'll basically spin around really fast and help propel the cell. It helps for movement. The fimbriae. So some cells will have lots of little appendages hanging off of it that almost kind of look like hairs, but they're not hairs. But they help attachment to surfaces. So it helps bacteria stick to something, which also increases their survivability. And lastly, conjugation pili. So this is one of my favorite ones, conjugation pili. They are going to aid with DNA transfer. So that's really beneficial for these bacteria cells, because once you understand how bacteria multiply and divide, how they reproduce and make babies, you'll know that when they do that, their offspring are genetically identical to the parent cells. So that doesn't create a whole lot of genetic diversity, and we'll talk about that in a later chapter. But conjugation pili are basically the formation of little tubes between one bacterial cell and a second bacterial cell. And then DNA can be transferred back and forth between them so they can share genetic material. All right, so this shows you a general schematic of a prokaryotic cell. So you can see the nucleoid region here has all the DNA just kind of in a little clump. It's not inside of a sac. It's free-flowing. You can see the little dots. Red dots are representing our ribosomes. You can see this plasma membrane as well as the cell wall. This shows you the cytoplasm, which is the fluid in here. This shows you the flagella. Now, some bacteria cells will only have one flagella. Some will have two. Some will have three. Some will have five. Some will have flagella all over their whole cells. Just depends on what kind of cell, what the species is. And you can see fimbriae as well covering the cell. This over here is a TEM image of that bacterial cell. So you can see the flagella here, all the fimbriae, and the internal contents. This is E. coli, by the way, 32,000 times magnification. So here's some common examples of prokaryotic organisms, and we just mentioned E. coli. Escherichia coli is the actual long name for that. So this one's probably pretty familiar to you. It causes the most UTIs, urinary tract infections, of any other microorganism. Also can cause foodborne illnesses that can make you have upset stomach, diarrhea, that fun stuff. However, E. coli is actually naturally found in your GI tract. It typically doesn't cause problems, but when it acts as an opportunistic pathogen, or if you have a particular strain that can cause disease, like traveler's diarrhea, there's hemorrhagic strains of E. coli, which are pretty nasty little buggers, but they can cause you to get sick. Next, streptococcus strains. The most common thing I can think of that you might be familiar with is the bacteria strains that cause strep throat. They're an example of a strep. Strep pyogenes, I believe, is the one that causes that. Now, other skin disorders can also be caused by streps or staphs. I know you've probably heard of Staph aureus, but that's definitely not the only staph out there. There's also several different strains of strep. Just to name a few, let's see, there's, of course, Staph aureus, there's Staphylococcus epidermidis, there's Streptococcus pyogenes, there's Streptococcus saprophyticus, and that's just to name a few. There's several other different ones. But any of those different strains or species can cause various skin disorders. So, for example, cellulitis, folliculitis, impetigo, and boils. All of those are skin infections, and typically they're caused by streps and staphs. One of my stepsons had impetigo about a year or two ago, and it was caused by Staph aureus. Very common in kids, because kids are gross. Y'all know. Apparently he was picking at an ant bite, and that's how it started. Anyway, don't pick your skin. Okay, so STDs can also be caused by prokaryotic organisms. STD stands for sexually transmitted disease. So chlamydia trachomatis, you can probably guess that causes chlamydia. Believe it or not, the current statistics are 1 in 4, I believe 25% of people will contract chlamydia before the age of 30. That's a lot. You want to know why? Because it is very, very, very likely that it will be asymptomatic. And so people have unprotected sex and spread it without even knowing that they have it, and then no one knows that they have it until probably it's a female going in for her annual checkup, and then she finds out she has chlamydia. Nowadays, it's kind of even scarier because it's becoming more common where people won't have to, or women won't have to go to the doctor to get birth control. You can just order it online now and not even ever see a doctor. And so that, as a microbiologist, that does kind of make me wonder how those numbers will look in the next 5 to 10 years, probably even higher. Anyway, make sure you don't have unprotected sex. Okay, next, Neisseria gonorrhea, also very, very common, caused by, or it causes gonorrhea, as you can tell by the name. Treponema pallidum, pretty common as well. This causes syphilis. Next, foodborne illnesses. We already mentioned E. coli, but there are others. Salmonella, Shigella, Vibrio, Listeria, Clostridium botulinum. That one causes botulism. Think canned food is where we're concerned about that one, because they have spores that can withstand very high temperatures. You have to be very careful with canning goods. But anyway, if you've ever been to the grocery store with your granny, she probably told you, oh, don't buy that can, it's dented. Well, this would be why, because that increases the chances that that can of food is no longer any good, and it contains this bacteria that can make you very sick. Now, all that, we've said a lot about bacteria being bad, but believe it or not, only, again, 1% of bacteria will make you sick. The rest of them are very good. They're found in dairy products, Lactobacillus, Streptococcus thermophilus, 2 of which are found in dairy products. Again, you have a whole flora, whole plethora of different bacteria that are inside of your body and on the outside of your body that are actually benefiting you. There's more bacteria cells that make up you than you cells. But we'll talk about one more nasty one because microbiology is my favorite subject. But this is MRSA. MRSA stands for methicillin-resistant Staphylococcus aureus. And essentially what that means is it's a staph aureus. That's the scientific name for that particular species of bacteria. It's staph aureus that is resistant to methicillin. Methicillin is an antibiotic and it does not work on staph aureus. And this particular staph is actually resistant to a lot of antibiotics. We do currently have antibiotics that do treat it, but it is much more concerning. It is becoming even more concerning as time moves on because it is being more and more common where MRSA is acquired in the community. So typically, before let's say the past several years, I'm not sure exactly how many, but previously to modern day, MRSA would be associated with hospitals. Like you go to the hospital and you might contract MRSA. Because, when you go to the hospital, people go to the hospital, it's because they're sick, right? So hospitals are, not exactly, they do a lot of cleaning and everything, but it is, it is possible. I don't want to say it's common, but it's not unheard of where people will contract bacterial infections because they went to a hospital. And that's what a hospital acquired infection is. And MRSA used to be known as a hospital-acquired infection. However, now it's found sometimes in the community. I actually watched a Frontline video about several different really nasty, scary bugs that people had contracted throughout the world. And there is one little girl, she was only 11 years old, who almost died from a MRSA infection because she scraped her knee at a playground and then she was picking at it. And so that MRSA basically caused sepsis of the blood. She wound up being put on ECMO, which then causes an infection of what was it? It was a stenotrophus bacteria. And long story short, she had to have a lung transplant, and this girl, it was really bad. So MRSA can be a pretty nasty bug. So it's a type of staph that's very resistant to antibiotics. It can be fatal. I don't know this lady personally, but there is one that worked at Memorial. I'm not sure if it was the Memorial in Savannah, but she died from a resistant staph. It was vancomycin resistant. So it's pretty scary. She was a hospital worker. She worked in the lab just like I used to at the Mayo Clinic, but she was at a Memorial Hospital and she died from contracting Staph aureus. Now, so how does this happen? So I keep mentioning antibiotic resistance. And so essentially, your body is full of germs and those germs are, good for the most part. Well, let's say one of them's not good and you go to the doctor. You get an antibiotic. You take the antibiotic. All right, so if you take that antibiotics are not always specific to what it is that you're infected with that's causing you to be ill. They're going to kill a lot of bacteria, not just what's making you sick. Now, if you don't take the antibiotic as you're supposed to, let's say you start feeling better within 5 days, typically antibiotics will be prescribed in a 10-day regimen, depending on the illness. But if you only took it for 5 days and you're feeling better, well, you may not have killed all of the bacteria that are, making you sick. That exposure to that antibiotic could actually cause them to become resistant. And now, as they grow and reproduce, you're now full of bacteria that are resistant to that antibiotic that you were supposed to take for 10 days. And then sometimes those bacteria that are resistant to that antibiotic can pass it on to other bacteria. Remember we talked about the conjugation pili. So now we got more than one type of bacteria in the body that is resistant to these antibiotics that are making you sick. And that just creates a big old mess. So here we see a MRSA infected foot. Lovely. All right, so that was prokaryotes. Let's move on to our eukaryotic cells. So eukaryotic cells are found in protists, fungi, plants, and animals like us. We're animals. They have a membrane-bound nucleus. So you think about it like here's our plasma membrane of our cell. Here is the nucleus, and the DNA is within it. So it's like almost like a water balloon within a water balloon, and there's where the DNA is. They're much larger than prokaryotes, and they have compartmentalized areas that we call organelles. Think of organelles like the organs of cells, just like your liver and your brain of your body. Your liver has digestive functions. Your brain has nervous system functions, helps you think, helps you remember stuff. They're examples of organs of your body. Organelles are organs of the cells that have specific functions for them. There's 4 categories of organelles: nucleus and ribosomes, endomembrane system organelles, energy-related organelles, and organelles of the cytoskeleton, and we'll go into each one. Before we do, let's look at a typical structure of an animal and plant cell. This one is an animal cell. This shows you a TEM, the transmission electron microscope image. But in red on the outside, we can see the plasma membrane. So we have here in the little dots, those are ribosomes. This is a mitochondria. We'll talk about what all these mean. You probably know that one though. Mitochondria is the powerhouse of the cell, right? Every time I talk to people, that's always, oh yeah, mitochondria is the powerhouse of the cell. I'm like, yeah. Everyone knows that one. Okay, here we have the nucleus. Excuse me. We have the nucleolus. We'll talk about what that is inside of it. Excuse me. We have centrioles, we have vesicles, we have rough and smooth endoplasmic reticulum, Golgi apparatus, we have lysosomes, yada, yada, yada. We'll talk about what all that means. Here's a typical plant cell. So one thing that's very prominent here that you see that you didn't see in the animal cells is this big old bubble here. This is a central vacuole. So animal cells have vacuoles too, but none that are this ginormous. This holds a lot of water and nutrients for plants. And that's really necessary because you got to think, plants, they can't really control when they're going to get water. They have to wait for someone to water them or wait for rain. So this stays full with water to act as a storage unit for it so that in periods of drought, they have food and water. Okay, the nucleus. The nucleus stores genetic information. And again, we said that's DNA. DNA is called chromatin when it's diffuse. Diffuse means very loosely coiled. Right? So it kind of looks like spaghetti in the nucleus. When cell division occurs, and we'll get to that in another chapter, DNA will condense to form really tight coils instead of being loosely coiled like that. It'll form really tight coils like this. And we call that chromosomes. So you might have heard that before, and so I wanted to elaborate as to what that meant and what the difference was. So chromatin versus chromosome, loosely coiled, tightly coiled. The next structure is the nucleolus. That is a region where ribosomal RNA is made. The nuclear envelope is a double membrane structure that encapsulates the nucleus, which is encompassing the DNA. All right, and then lastly, the nuclear pores. They are little tiny holes, you can see them all throughout, that allows passage in and out of the nucleus. Not very many things go in and out of the nucleus, but some things need to, like mRNA, and we'll discuss that at a later date. Okay, next, ribosomes. They carry out protein synthesis. And again, that's super important, super, super important. Ribosomes, protein synthesis. They build proteins. What do they build them from? Amino acids. Remember, they're the building blocks of proteins. But ribosomes are found in both prokaryotes and eukaryotes. They're made up of 2 subunits, a small one and a big one, and they basically sandwich together. They receive mRNA, which is messenger RNA, and that is going to give them instructions on which amino acids to to put together to make a protein or a polypeptide. So maybe they'll get instructions to add glutamate, and then they'll get instructions to add the next amino acid, which is alanine, and then maybe the next one will be valine. That mRNA is going to tell them what amino acids to string together to make that polypeptide, aka protein. In eukaryotes, some ribosomes are free in the cytoplasm, but a lot of them are attached to the endoplasmic reticulum. So that's not the case for prokaryotes, because prokaryotes don't have an endoplasmic reticulum. Now, endoplasmic reticulum, we're going to get to on the next slide. It's part of the endomembrane system. Okay, so the endomembrane system includes the nuclear envelope, the ER or endoplasmic reticulum, the Golgi apparatus, and a lot of vesicles. But all of these different organelles are going to help compartmentalize the cell. And when I say compartmentalize, I mean to organize it into little tiny compartments. And those compartments help to restrict reactions from going on, or it helps prevent reactions from taking place in all parts of the cell. We want it to only happen in specific parts of the cell. So think about it as a means of organizing chemical reactions. Just like if you have a, I don't know, let's see, like a makeup bag that's very, very organized, and you put your little makeup brushes in one part, and then you put your eyeshadow in another. And that's, that's what I mean by compartmentalizing. It's organized. Things are supposed to happen in certain spots. I'm trying to think of a male version of that example. Let's see. Ooh, here's a good one, a toolbox. So you put your wrenches in one part of the toolbox, and you put your nuts and bolts in another part, and your drill goes over here. You're compartmentalizing things because certain things go in certain spots. So that's kind of the goal here of our endomembrane system, to help compartmentalize the cell. Certain things need to happen in certain areas. Okay, so the endoplasmic reticulum I'm going to refer to as the ER to save my sanity and my voice. But the ER can be rough or smooth. The ER in general is pretty complicated. It has a lot of convoluted membranous channels and sacs, and basically it kind of almost looks like a very complex stack of pancakes, sort of. But it's basically a lot of membranes. It's again, can be rough or smooth. The rough ER is called rough because if you look at the picture here, it's studded with these little red dots, and those red dots are representing ribosomes, and that's why it's called the rough ER. It looks rough. But the smooth ER, on the other hand, is smooth. Okay, so rough ER, smooth ER. Rough ER has the ribosomes, and smooth ER does not. So that might give you a little hint then to what the function is of the rough ER. It aids in helping to build proteins because ribosomes help make proteins. So these ribosomes will produce proteins that then the rough ER can transport to the next organelle, which is the Golgi apparatus. We'll get to that one in a second. The smooth ER, no ribosomes, no protein production. Instead, here, the smooth ER synthesizes lipids, fats, oils, phospholipids, steroids that can then be transported to the Golgi apparatus as well. So in the Golgi apparatus also looks like a stack of flattened little sacs. They all kind of look like a whole bunch of pancakes to me. But you can think of this like the post office of the cell, because the Golgi apparatus will receive the things that the rough and smooth ER made. And it will modify them and package them, just like the post office does. And then it will sort and repackage them to a new destination. All right, so if you take something to the post office, they put a label on it. That's them modifying it. They might put it in a bigger box, repackage it, and then they're going to send it somewhere else. So that's why I say they're kind of like the post office of the cell, the Golgi apparatus. Next is lysosomes. So these are little vesicles that are made up of a membrane that digest stuff. Think of them like the garbage disposal of the cell. They digest the trash. And so that even though that seems like a relatively important job, it actually really is important. So there is a lysosomal disorder called Tay-Sachs disease that can cause issues with the nervous system. Trash getting built up in the cells is never good, but there's no patients that have lived past the age of 4 with that disease. Okay, so here is a summary of the endomembrane system. So we started off with a nuclear envelope, which surrounds the nucleus. Then we have the rough ER, which is studded with ribosomes. Over here we have the smooth ER, which is not. It functions to make lipids. This one functions to help make proteins, both of which can pass in vesicles to the Golgi apparatus. Over here you can see it's passing vesicles that contain lipids to the Golgi apparatus, and then those vesicles will be packaged and modified in the Golgi apparatus, which is in the blue, and then after that it can be sent off, you can see that here, somewhere else to a new destination. Over here, these are where the lipids were produced by the smooth ER, and then they can be modified, packaged, and can ultimately be sent to another destination via the Golgi apparatus. They can merge with lysosomes like this one, like this one, and become destroyed by the lysosomal contents. Lots of digestive enzymes in there. Sometimes things will just be sent outside of the cell. Sometimes they'll just be sent to a different part of the cell. Just depends on what the cell's needs are. Okay, vacuoles. So we've mentioned those a few times, but again, they're also membranous sacs. They're basically used for transport of stuff. They can aid in digestion whenever they fuse with a lysosome. So that's what we saw in the previous picture with lysosome and vacuole. The lysosome will dump its contents into the vacuole, making it very acidic and degrade whatever's inside of the vacuole. All right. Vacuoles can also be used as storage. So pigments can be stored within vacuoles within plants. Fat can be stored within adipocytes. Adipocytes are basically fat cells. Adipo means fat. Cyte means cell. All right, so here's a couple pictures of vacuoles. This is a contractile vacuole of a paramecium. Parameciums can use these in a way to somewhat help propel themselves, and it also has something to. It also has it's a means of gathering food as well for them. Here we have a very large vacuole in a plant cell, which I showed you several slides back, but this is a TEM image of that same thing, transmission electron microscope. Okay, so moving to organelles, the first one is the chloroplast. All right, oops. The chloroplasts serve as a site of photosynthesis. And as we should recall from the very beginning of the class, photosynthesis is the process of capturing solar energy and converting it to food. So plants, animals, and some bacteria are capable of photosynthesis. Again, they use light energy to drive their metabolism, their cellular machinery. They're bound by a double membrane. They have an inner membrane and an outer membrane. The outer membrane is seen here. It's like on the outside of the organelle. And then on the inside membrane, it is organized into little discs. You can see all these various little discs. We call those disc shapes thylakoids. And then the discs, the thylakoids, are stacked, and those stacks are called grana. Okay, so each individual little disc is called a thylakoid, and the stack of the disc is called the grana. They are suspended inside of the stroma. The stroma is the liquid. It is just like the cytoplasm is for the cell. So again, let me draw up here. Within the cell, it's full of fluid, which is mostly water. We call that cytoplasm. The fluid within the chloroplast is called the stroma. Same thing, different name because it's in a different spot. Now, these chloroplasts are green, and that's because they are packed with chlorophyll, which is a green pigment. And that's why plants are green. And last point, they have their own DNA. Next, mitochondria. Mitochondria are found in both. I want to make that very clear because I've seen lots of students get confused and think that plants only have only have chloroplasts, but they don't. They have both. Animals, however, only have mitochondria. We don't have chloroplasts. We can't photosynthesize. So anyway, mitochondria are involved in a process called cellular respiration, which we'll get to in another chapter. So it's basically our form of getting food. Like we said, with plants, they can photosynthesize. They can get food from the sun to get energy to make energy for them. But in animals or organisms that can't photosynthesize, we perform cellular respiration in the mitochondria, where we break down food, such as carbohydrates primarily, to produce energy. Our form of energy is called ATP. You need to know that. Adenosine triphosphate. Adenosine triphosphate, ATP. Now, mitochondria are also surrounded by a double membrane. You can see we have one on the outside that surrounds the whole thing, and the inside that's very convoluted. It doesn't form stacks of thylakoids like it did in plants, but it is still pretty convoluted. These intermembrane folds are called cristae. You need to know that. And then the fluid in the mitochondria is called the matrix. Just like the cytoplasm in the cell, the matrix is the fluid in the mitochondria. All right, we're getting towards the end, you guys. The cytoskeleton. The cytoskeleton is a network of protein filaments and tubes that are found in eukaryotic cells that help to maintain the shape. They assist in movement of cells and organelles and make internal transport possible. There's 3 different types: actin filaments, intermediate filaments, and microtubules. And all of these will assemble or disassemble as needed. Let's start with actin filaments. Actin filaments are really thin. They kind of look like a pearl necklace that's been twisted. And they form a very dense web just underneath the plasma membrane of cells to help maintain their shape and also to help sometimes with movement. So a great example of that are cells that partake in movement by pseudopods like amoebas. So this is an example of an amoeba here. It's a single cellular protist. A pseudopod, if we break that down, pseudo means false, pod means foot. Podiatrists, they study feet. But they form these false feet because they're not real feet, but they act kind of like feet. So you can see one's here, there. But these little pseudopods, they're basically forming due to the plasma membrane extensions. So pretend this is an amoeba cell. It's just a single cell. Now, if this cell wants to go this direction to the right, it's going to have a lot of actin filaments. You can see they're already all over there in red. But that's going to form a lot of them. They're going to start building them on whatever side they want to go to. That's going to cause that plasma membrane to stretch out and reach. And it does. And it reaches some more. And so as it finishes reaching, those actin filaments will then start to depolymerize eventually once it's ready. But in response to this cellular extension, we're going to see retraction on this end. So it almost kind of sort of reminds me of an inchworm. We're stretching out this way and we're retracting at this end. And that's due to actin filaments polymerizing, or that means to build actin, making that stretch. and depolymerizing, which means breaking it down. This is also another important component of muscle contraction. So our muscle filaments are made up of actin and myosin, and they basically, to make a long story short, they slide across one another to shorten the muscle to produce force. Intermediate filaments. So these are in an intermediate size between actin filaments and microtubules, and they look rope-like. Their function is to support the nuclear envelope. They aid in cell-to-cell junctions, like holding skin cells together, keeping them watertight. And, some other examples are the protein keratin. Keratin is a protein that helps give strength to skin cells. Your hair is made up of keratin as well. You might have heard of keratin shampoos help restore that very strong keratin protein. Your nails as well. Next, microtubules. They are tiny tubes. Exactly, right? Microtubules. They're little tiny tubes. So you can see the tiny tube here. Now, that tiny tube interacts with motor proteins or motor molecules. Some examples are kinesin and dynein. And kinesin and dynein have the capability to aid in cellular movement of organelles and other different molecules. So here again, I'm going to use the mouse. I think it's easier to see. But you see your microtubule here. This is a protein called kinesin. This is our kinesin receptor, which is going to bind to a vesicle that contains contents. This could be an entire organelle. This could just be a protein. It could be anything. It can help move stuff. And literally what kinesin does is it attaches its little feet here to this microtubule, and it holds on to whatever's in this vesicle, and literally it looks like it walks down this microtubule just like you would be walking down the street. It's really crazy, isn't it? I think it's so cool. I love molecular biology, but literally it just looks like it walks down this little microtubule and takes it from point A to point B. All right, next, centrioles. They are made up of 9 sets of these microtubules in triplets. So you can see triplet microtubules here, there, there, there. And if you count them all up, there's 9. 2 of them lie in the center at right angles. And these are found in animal cells, but they're not in plant cells. They're involved in the process of mitosis, and we'll get to that in another chapter. But it's essentially division of chromosomes during cellular duplication. Next, cilia and flagella are 2 potential extensions that cells can have. Cilia are hair-like projections. So this would be a great example of cilia. So all of these dark things here, these are nuclei of various cells. So this is many, many cells, and at what we call the basal membrane here, we have lots of little hairs that are called cilia. So this cilia can involve the ability of cells to move stuff. A great example would be in the upper respiratory tract, like the larynx, the windpipe. It's lined with, or the trachea actually. The trachea is lined with these cells, and the trachea is the tube that's right above the lungs. And so we want to avoid getting goo and gunk and snot and mucus into our lungs, right? We don't want fluid in our lungs. That causes pneumonia. So these little hairs beat essentially back and forth, kind of like they're rowing a boat. They're going back and forth as you breathe in and out. And that helps to push any mucus that collects in the trachea back up. So then you'll either, spit it out, or you'll cough it up, you'll, blow your nose, or you'll, swallow it. Something besides letting it get to your lungs. All right, lastly, flagella. So flagella are long tails. We've seen those before with prokaryotes. A great example are sperm cells. They have these really long flagellas that they can move back and forth to propel them forward to go on their long travels. Okay, 4.5 is outside of the eukaryotic cell. We'll first discuss the things that are relevant to plant cells. Plants have primary and secondary cell walls. Primary cell walls are made up of cellulose primarily. Secondary cell walls are primarily made up of lignin, and they're found in woody plants. So not all plants, just the woody ones. But it gives them extra strength to stay their shape and to get very tall. Plasmodesmata are little holes, little channels between different cells. So here we have cell number 1 and cell number 2, and this little channel allows them to share the contents of their cytoplasm. Now let's talk about animal cells, so exterior cell surface in animals. So animals do not have a cell wall, no cell wall. They do, however, have what we call an ECM. This is the extracellular matrix. Extracellular matrix. Extra means outside, and cell means cell. So it's the space literally outside of the cells, in between cells. And in between cells, there's a lot of meshwork of fibrous proteins. When I say fibrous, I mean they look like fibers, strands, as well as some polysaccharides, carbohydrates. Some examples, collagen and elastin are very well known to be in the ECM. Collagen and elastin are proteins that help cells stick together really well and help with elasticity. And as we get older, collagen and elastin production slows down, and that's what causes us to get wrinkles and why our bodies just don't bounce back the way they would when we're older if we're having children versus, when someone's really young and has children. If you pinch your skin right now and you're relatively young, it's going to pop right back into place. But as you get older and collagen and elastin have been broken down and not replenished very well, it's not going to pop right back into place. But anyway, the matrix, that ECM can vary. It can be very flexible. In cartilage, you have very, very hard extracellular matrix when it's in bone, and so on. And here's a depiction of that. So here we have the inside of the cell. We have the cell membrane. We see some proteins as well. And then on the outside, you can see some polysaccharides. You see collagen and elastin fiber. And there's some other things that are in there too. Those are just some of the most well-known ones. All right. Lastly, some junctions between cells. This is showing you 3 of them. We'll discuss them in a little more detail, but I just kind of wanted to show you a general picture just so we understand what's being shown. This is showing you a small section of cell number 1 and cell number 2, and then the junction between. Same thing here, cell 1, cell 2, and then the junction between. This is showing you plasma membrane of cell 1 and cell 2, and the junction between. So the first one was an adhesion junction. So they have a internal cytoplasmic plaques that are joined by intercellular filaments here. They're very sturdy, but flexible. Tight junctions. They form an impermeable barrier. Water will not even pass. So you can again see plasma membrane number 1, number 2 is on the other side, and you see all of these tight junction proteins all throughout forming a very tight barrier that is even not subjected to water passing through. And lastly, gap junction. So here again we have cell membrane number 1, cell membrane number 2, and we can see these membrane channels. And those are called gap junctions. It allows communication between these 2 cells. They're almost sort of sharing their plasma membrane, or their cytoplasm. Sort of similar to plasmodesmata in plants. All right, that sums up chapter 4, you guys. I hope you enjoyed it. Have a good night.